Hi, Dr. Zhou. We are JiaXu, Boonyee, Gerald, Benjamin, and Chunsiang. The topic we'll be doing is the production of L-Alanine. What is L-Alanine? It is an alpha amino acid that is used in the biosynthesis of proteins. It contains an amine group and a carboxylic acid group, both attached to the central carbon atom, which also carries a methyl group side chain. It is widely used for protein formation, as well as an important source of energy for muscles and central nervous system. Its molecular formula is C3H7NO2. We'll be discussing two methods used to produce L-alanine. The two methods are metabolically engineered E. coli and chemical synthesis, also known as tracker amino acid synthesis. Firstly, we'll be discussing the metabolically engineered E. coli. The E. coli transforms glucose into L-alanine. The glucose is first oxidized by NAD plus and ATP molecules to produce NADH, two net ATP molecules, and two pyruvate molecules in the E. coli. Pyruvate is then being reduced into L-alanine molecules with the help of alanine dehydrogenase enzyme and NADH molecules. My groupmate Gerald will now take over.
So how are these E. coli metabolically engineered? Using PCR, which is known as polymerase chain reaction, to duplicate and amplify ALAD genes for synthesis of pyruvate. Use restriction enzymes to cut specific ALAD genes from the DNA strand and the MCS of plasmid, which is the vector we are using. Then use ligase to bind the gene of interest with the vector. Using the expression vector, we can transform the E. coli cells. Allow the transformed E. coli cells to grow and duplicate. These cells grow best at body temperature, around 37.4 degrees Celsius, with thick chicken broth for nutrients and energy, an aerobic environment such as a culture flask in the laboratory. Under ideal conditions, individual E. coli cells can double every 20 minutes. At that rate, it will be possible to produce a million E. coli cells from one parent cell within 7 hours. So this is the overview of the glucose synthesis in the E. coli. The pathway to produce L-alanine is underlined in red. The glucose is being reduced to pyruvate, and then with the help of ALAD, it's being oxidized to L-alanine. There are also other byproducts which are underlined in green, such as succinate, ethanol, and acetate. So how do we increase the yield of L-alanine? There are many byproducts produced during the synthesis of L-alanine, such as D and L lactate, carbon dioxide, hydrogen gas, ethanol acetate, succinate, and D-alanine. We can lessen yield of these additional byproducts by deleting certain enzymes required to form those products. The more important ones are MGSA and LDHA to reduce the production of D and L lactate. ALR and DADX racemase genes to reduce the amount of L-alanine being converted into D-alanine. There are also other enzymes which require the deletion which are in the slides. This will help to drastically increase the overall yield of L-alanine. Now I will be passing it on to Chunsiang.
Our second method of production is chemical synthesis, also known as the tracker amino acid synthesis. L-alanine can be synthesized from an aldehyde. The process is condensation of the aldehyde with ammonium chloride in the presence of hydrogen cyanide, forming an alpha-aminonitrile, which is then hydrolyzed to obtain the desired amino acid. The original tracker reaction combines acetaldehyde, ammonia, and hydrogen cyanide to give alpha-alanine after hydrolysis. So the acetaldehyde will react with ammonium chloride to give the amine, and then it will, in the presence of hydrogen cyanide, it will produce aminonitrile, and then it will undergo hydrolysis to produce alpha-alanine. Using this method, it will produce alpha-alanine, meaning that there will be both D-alpha-alanine and L-alpha-alanine being produced. More money is required to obtain an amino acid racemase to convert D-alanine to L-alanine to get the best percentage yield of L-alanine from acetaldehyde. The strength of both methods are that they can undergo very large-scale production. However, metabolically engineered E. coli requires many steps and materials for the production of L-alanine. Not all E. coli might be successfully transformed, so this affects the total yield of L-alanine. The weakness of chemical synthesis is that the produced amino acids are D and L isomers, and additional optical resolution step is needed in order to obtain bioactive L isomers. And this additional step has high costs. Based on economic profiting, using metabolically engineered E. coli is more suitable. Based on consistency and simplicity, using chemical synthesis is more suitable. Now I will pass it on to Benjamin.
For my part, I'll talk about the business plans of L-Alanine. Before I specifically go into the business plans, I would like to talk about the users of L-Alanine, because this will help me figure out the target audience of producing L-Alanine. For users, I split it into two parts, such as the health benefits of L-Alanine and the way L-Alanine boosts one's performance. Going into the health benefits of L-Alanine helps people who are suffering from hypoglycemia, which is a condition caused by very low level of blood sugar. It is also used as a supplement to compensate for those with insufficient L-alanine in the body. It is also used to help treat benign prostatic hyperplasia, also known as the prostate gland enlargement. It helps to support muscle development, as taking L-alanine prior to a big workout can increase energy levels and the duration of the exercise. It helps to support immune system by producing antibodies. Now going into the performance of L-alanine. L-alanine helps to relieve muscle fatigue, which is significantly useful for older people, as L-alanine keeps toxins from being released during the breakdown of muscles. As a result, this allows for faster recovery. It also helps to improve one's fitness to prevent cardiovascular diseases such as stroke. Therefore, our target consumers will be people with active lifestyles, as L-alanine helps with the release of blood glucose, which increases performance, helps build muscles, which help consumers become fitter, and it also reduces fatigue, so consumers can increase the intensity of training. It also helps people with diseases such as it can elevate low blood glucose, also known as hypoglycemia. For elderly, it helps to relieve fatigue and strengthen muscles. As for safety concerns of L-alanine, it seems to be safe when used appropriately for a short time. As long-term consumption of L-alanine increases blood sugar level, it can result in high blood sugar. This is because liver absorbs the L-alanine introduced that will be converted back to pyruvate and glucose eventually through gluconeogenesis. As for side effects of L-alanine, it has not been reported in studies of any possible side effects resulting from consuming L-alanine.
So moving on, I will talk about the market for L-alanine. Currently, there are already a large number of pure L-alanine products being sold as supplements. Brands include Monteith, Source Naturals, and many more are available online, where most of these supplements are targeted at improving fitness. Talking about the demand and popularity of L-alanine, Singapore is experiencing an aging population. As a result, the government has expected the total number of citizens aged above 65 to hit 900,000 by 2030. This results in supplements being more popular in demand amongst elderly to boost health conditions. The demand for L-alanine supplements will also be steadily increasing due to the aging population. Other than the aging population, Singaporean lifestyles also play a part in the demand and popularity of L-alanine. This is because the workload and work environments of Singaporeans usually leave them having less time for exercise. This results in people relying more on supplements to improve health. And for the last condition of demand and popularity of L-alanine, I will talk about athletes. L-Alanine supplements are popular among athletes to improve sport performances. This is because L-Alanine can also help build muscle, which helps consumers become fitter. We have come to the end of our presentation.